Hi, everyone. For those that don't know me, my name is Colleen Burge, and I run the Shellfish Health Lab. And today, I'm really excited to introduce Dr. Yamini Venkataraman, and I probably just murdered your last name, as our seminar speaker. Yamini received her BS in Biology and BA in Environmental Policy from UC San Diego, followed by her PhD from the University of Washington School of Aquatic and Fishery Sciences. Her doctoral research focused on the effects of ocean acidification on Pacific and Eastern oysters using epigenetic approaches. This is also when I first got to know Yamini, through our shared interest in shellfish and omics research, and when she was an excellent TA for my marine disease ecology course at Friday Harbor Labs. Following Yamini's PhD, she was a postdoctoral scholar at Woods Hole Oceanographic Institute, where she expanded her research to examine how environmental stressors affects a range of marine organisms, including killifish, Caribbean corals, and green crabs. Yamini recently joined the faculty at Santa Clara University and is assistant professor, where she is building her research program and actively seeking new collaborations, so potentially all of you. And we're very excited to have her here today to share her work on You Win Some, You Lose Some, how phenotypic plasticity shapes resilience to climate change. So please join me in welcoming Yamini.
Thanks, Colleen, and thanks to all of you for coming today. I have visited Bodega a couple times in the past year, mainly to come and get green crabs that were collected for me and steal some of your filtered seawater and drive 2.5 hours south. So I'm excited to be in your buildings that aren't your seawater facility for the first time, and to talk to you all about some of the work that forms the foundation of my lab at Santa Clara University, where, like Colleen mentioned, I'm finishing up my first year as an assistant professor. So to start, this is not news to any of you, but the ocean covers about 70% of the planet and is home to staggering amounts of biodiversity. And this biodiversity is important ecologically, economically, culturally, socially, and of course, this biodiversity is threatened by climate change. To move forward. Okay, great. Climate change manifests in several different ways, but the ways that I think about climate change impacts are ocean warming and ocean acidification. So this figure right here shows a sea surface temperature anomaly that we all know as the blob that formed off the Pacific coast in 2014, as well as the formation of a similar sea surface temperature anomaly forming off the Pacific coast in September of 2025. And I'm sure you could look around and find a map of a sea surface anomaly that's developing now as we barrel into potentially a monster El Nino this fall and winter. Right, so I think about ocean warming and these anomalous heat waves. I also think a lot about ocean acidification. So this map I really like, and it's from one of the IPCC reports. And I like it for two reasons. One, this map shows projected change in pH across the ocean based off of the most extreme emissions scenario, but it also overlays that change in pH with the location of mollusk and crustacean fisheries, as well as cold and warm water corals. So showing us where we're expected to see these large swings in pH conditions and how they overlap with organisms that we care about. Right, so thinking about these impacts separately or together, right? These stressors can lead to losses of biodiversity that we care about, the poster child of which I feel oftentimes in the media is a bleached coral reef, but it can also lead to alterations in biodiversity like non-indigenous species introductions. So the thing that I think about in this context, the thing that keeps me up at night, is trying to understand why some organisms are resilient to climate stressors and why some are susceptible to climate stressors, right? Why do some organisms win? Why do some lose? And what are the mechanisms that underpin these responses to climate change? So when we first start to think about mechanisms, we can start with genetics, right? Broadly, we can consider an individual organism's complete set of genetic material or their genotype. And And in this, every population likely has some variation of genotype, as depicted by this oyster population of several colors, where each color corresponds to a different genotype. Genotype also influences phenotype, which are the set of characteristics that we observe in an organism. Stressors such as ocean warming or acidification may remove less tolerant genotypes from a population, right? So we had a population with some diverse genotypes, stress event rolled through, now we're left with only some of those genotypes remaining. But of course, genotype itself isn't the only thing that dictates species resilience or susceptibility to stressors, right? We have phenotypic plasticity, which is the ability of one genotype to produce more than one phenotype in response to environmental variation, with acclimation being one kind of plastic response, which is generally the process of adjusting to a new condition. So in addition to genotype, plasticity becomes this important factor to consider as we think about how organisms respond to climate change. So I have a background in both physiology and molecular biology and functional genomics. I like to combine these two very related fields of study to get a holistic understanding of how species respond to climate change. And in some ways, I liken this to being like a car mechanic, right? So let's say you have a car and your car won't run. Great, it's good to know that your car won't run, but why won't it run? Is it because your battery's dead, you have a flat tire, or is it because your car is on fire, right? Those are very different scenarios, and understanding the why of why can't my car run is important for then solving your problem and getting your car to run again, right? So by integrating physiology and molecular biology, we can get at the why, we can get at that mechanism, so we can use the information that we have to manage populations of animals that we care about. So, what are some of the tools in my molecular toolbox? I teach intro bio, so when I talk to my students about DNA, I say DNA is like a cookbook filled with various recipes that an organism needs to stay alive. Those individual recipes are our genes, right? But this cookbook that you have isn't a brand new one. It's a used cookbook. So there are pages of that cookbook that are covered in highlights or whiteouts, or perhaps there's some notes scribbled in the margins. Those annotations don't change the recipes themselves, but they can change the way those recipes are interpreted, right? So this manifests biologically as epigenetic modifications. One of the ones that I'm interested in is DNA methylation. So DNA methylation is the addition of a methyl group that, I get this pointer work here, haha, yes, the CH3 group that's attached to a cytosine next to a guanine base. That little methylation lollipop on the DNA doesn't change the underlying DNA sequence, but it changes how the cell interprets or expresses that gene, right? So kind of similar to that analogy of having a used cookbook with some highlights or whiteouts, right? So we've got our DNA, we've got our genetic sequence, we've got a methylation pattern on our DNA sequence, and that methylation pattern may be able to be inherited the same way DNA is inherited from parents to offspring. So in non-stressful conditions, we would have DNA with some sort of baseline methylation pattern. That DNA gets transcribed into RNA, which then gets translated into proteins, central dogma of biology. And through various protein substrate binding processes, we get smaller molecules like metabolites and lipids. And all together, these molecules shape the physiological phenotype of an organism, which in this case, let's say, is the survival of these 4 oysters. When faced with stressful conditions like a marine heat wave event or ocean acidification, that baseline methylation pattern changes. That change in DNA methylation then impacts the genes that are expressed, right? So, oh, hang on, can I go back? Yes. So previously we had 3 different colors of transcripts being expressed. Now with a difference in methylation patterns, we're seeing a reduction in that transcriptional noise. So only a couple different transcripts are being expressed. That leads to a change in our protein abundance, a change in our metabolite and lipid abundance, so on and so forth, which all may radiate up to a change in physiological phenotype. So where previously we had 4 oysters that were surviving, now we only have 2. So one example that I like to use of how we can go from environment to molecules up to physiological phenotype is the sex determination of the European sea bass. So normally aromatase, which is a protein, will convert male hormones to female hormones, which is necessary for ovarian development. But researchers have found that in warmer ocean conditions, the aromatase gene actually has a different DNA methylation pattern on it, such that we get lower abundance of that aromatase protein. So then we get less male hormone converting to female hormone, and that radiates up to a skew in our sex ratios in our European sea bass populations. Right? So I'd just like to use this example to show why it's important to study the mechanisms of climate change physiology. Right? Changes to sex determination physiology can have cascading impacts on reproduction and population fitness in organisms like the sea bass. So today, I'm going to talk to you about a couple of systems that I work in, the Pacific oyster Crustacea dragus and the European green crab Carcinus manus. And in both of these systems, we're going to talk about, again, the question that keeps me up at night: how does plasticity shape species resilience to environmental stressors? So we're first going to start by talking about oysters. And broadly, I use pH manipulation experiments to connect changes in the environment with DNA methylation and physiology to understand the mechanisms that govern intergenerational plasticity. So apart from being interesting model organisms for organismal biology, oysters have ecological, economic, and social relevance. Right? They create oyster reefs, which protect our shorelines from erosion. Oyster aquaculture is a cornerstone of coastal communities, much like the coastal communities out here. And then shellfish broadly have cultural importance for indigenous groups. Okay. And then, of course, these ecological, economic, and social benefits can be threatened by climate change, specifically ocean acidification. So when I think about the impacts of ocean acidification, I think about them in 2 broad but connected buckets. The first thing I think about when it comes to ocean acidification impact on shellfish is just that with lower pH, it's just more difficult for these organisms to maintain homeostasis. Ocean acidification also leads to lower availability of carbonate ions, which is important for organisms like mollusks because they use these carbonate ions as shell building blocks for their shells and other calcified structures. Right? So because of this kind of 1-2 punch on mollusks, there is actually a wealth of research looking at how ocean acidification impacts shellfish. But the little pocket of research that I was most interested in when I started working in this system is the research looking at carryover effects. Carryover effects, or particularly intergenerational carryover effects, it's this idea that parental experience impacts offspring experience. So if parental oysters are exposed to ocean acidification conditions, that can then shape how their offspring respond to the same conditions. So we could think about positive carryover effects, where if mom and dad oyster are exposed to low pH conditions, we see larvae, those offspring, may have higher survival, they might be larger, or they may be faster growing. We also see literature in some species that shows negative carryover effects. Parental exposure to low pH can lead to smaller larvae, lower growing larvae, and poorer survival. And so while there is a wealth of research exploring these carryover effects in several oyster species, we don't quite know why we see certain carryover effects manifesting, right? What are the mechanisms underpinning these carryover effects is a little bit unclear. So given this information, the goal for my PhD when I first started working with oysters was to investigate if DNA methylation could be a mechanism that was mediating some of the carryover effects that we see in shellfish species in response to ocean acidification. So going into this, I had a couple of hypotheses. The first of which was that low pH would alter gonad methylation patterns, right? Where gonad is our reproductive tissue. That differential methylation would then be associated with physiological changes in the organism. And then finally, offspring would inherit environmentally sensitive methylation that would then influence how they responded to low pH conditions themselves. So to test these hypotheses, I worked with hatchery partners at Pacific Hybrid in Washington State to conduct a 7-week pH exposure experiment, where we had some oysters exposed to low pH conditions of about 7.3, and then some oysters exposed to ambient pH conditions of 7.8. We picked these conditions because they were ecologically relevant for Puget Sound, where we were conducting these experiments. After that 7-week pH exposure, we looked at gonad maturation in our oysters, and we also characterized the female gonad methylome. After that 7-week pH exposure, all of our remaining adult oysters were transferred to ambient pH conditions for 12 weeks, where at the end of those 12 weeks, these underwent a period of reproductive conditioning. And then finally, we strip spawned those oysters, and we measured larval survival in ambient pH 18 hours after fertilization. So let's start by just talking about this adult 7-week exposure experiment. So I'm going to talk to you about some gonad maturation data, but I wanted to show you what that data looked like. So these are histology slides of oyster gonads. These at the top are developing females. We know they're female because, if I can get a better view of this, right here, these kind of like larger purple dots are the start of developing oocytes or egg cells. We also saw some mature females. We know they're mature because that oocyte is a lot larger. And then we saw mature males, where all of those tiny purple dots were individual sperm. And at the end of our 7-week pH exposure, we actually saw no differences in reproductive maturation between our 2 pH treatments, right? So no difference in maturation stages, and no difference in maturation stages by sex, or no difference in sex ratios. So this was interesting for us to see that low pH didn't actually seem to have any sort of impact on reproductive development. Our next step was to characterize the female gonad methylome. And the reason why we chose female oysters as opposed to looking at male and female oysters is because eggs are a lot more of an energetic investment than sperm. And we figured that if we were going to start exploring these potential mechanisms driving carryover effects, the more energetically costly gamete would be a good place to start. So again, I want to start by showing you what DNA methylation data looks like. So this is DNA methylation data, where each row is methylation data for an individual oyster. At the top, I have our oysters exposed to ambient pH, and at the bottom, our oysters exposed to low pH. And I don't know about you, but if I look at this, I don't see any broad sweeping differences between those 2 treatments, right? So what we need to do is drill down to the individual cytosines, or CpG loci, where methylation occurs, and look for differences at those individual sites. So when we drilled down, we can see CpG loci with no methylation difference. So I know this has no methylation difference because the height of the bar corresponds to how methylated a specific position is. And if we compare our ambient and low pH, the height of those bars looks about the same. We can also see places with methylation differences. So on the left, this is what we call hypomethylation, where our treatment, our low pH treatment, has lower levels of DNA methylation than our ambient treatment. And on the right is the opposite. We have hypermethylation, where that low pH treatment has higher levels of DNA methylation than the ambient pH treatment. And overall, we saw significant differences in methylation in almost 1,300 CpG loci. And this is what we'll refer to as our differentially methylated loci, or DML. So now that we know there are some differences in DNA methylation, the next step is to figure out where those DML are located in the oyster genome. So I'm going to show you a stacked bar plot, where on the left, I have the percent of methylated sites, and on the right, I have a color key that tells you what genome feature that DML was found in. So the top three, exon untranslated regions, coding sequences, and introns, that makes up our gene bodies. Apart from our genes, we have flanking regions. So upstream flanks are the area before the gene starts. Downstream flanks are the area after the gene is ended. We have long non-coding RNA, transposable elements, which are parts of the genome that can move around, and then intergenic regions. So that's just everything else that isn't categorized as one of the other things that I've mentioned. So I've got our stacked bar for all the places DNA methylation can occur, so all CpG loci across the Pacific oyster genome. And on the right, I have those almost 1,300 DML. And when I look at this bar plot, the big takeaway for me is that we have a lot of those DML concentrated in those gene bodies, right? And because we're seeing a higher proportion of them in those gene bodies, that suggests to us that they have a role in gene activity. Right? So the final step in our kind of analysis pathway is to understand, okay, we identified DML, we know that a lot of them are in higher proportion in gene bodies. What do those genes with DML actually do? So I'm going to walk through the potential influence of these DML on physiology in the oyster by talking about two main groups of biological processes related to the genes with differentially methylated loci. So a lot of genes with differentially methylated loci were involved in the cilium and flagellar motility biological process. So cilium and flagellar motility, right, those are like all the little hairs on your cell, as well as microtubules that may move parts of the cell around, may be responsible for shaping tissue structure, right? And so some genes involved in these processes that had high concentrations of differentially methylated loci are genes like dynein, right? And so the implication of having a lot of differentially methylated loci in genes like dynein is that perhaps methylation is helping regulate microtubule movement in response to low pH to potentially promote gonad growth over somatic tissue at low pH in our gonad organ. Right? So dynein being differentially methylated may alter how it's functioning to promote gonad growth. Similarly, another group where we saw a lot of genes with high levels of differential methylation is this like broad category of stress response and gonad development. And these included a lot of genes that in the literature are both responsible for low pH stress response and gonad development across pH conditions. So for us, the implication is that genes with these differentially methylated loci that are involved in this process, again, may be responsible for promoting oogenesis or the creation of oocytes, eggs, in stressful conditions, right? So regulation of all these genes by differential methylation may have promoted that gonad growth in low pH to the point where we didn't see any differences in reproductive development between our pH conditions. So we know what happened with the adults. The remaining oysters were then reared and spawned in ambient pH conditions, and then we measured larval survival. So what happened with the larvae? I'm going to show you a box plot where I've got the proportion of live larvae on the y-axis and the treatment that the female oysters were exposed to on the x-axis, with ambient pH on the left and low pH on the right. And we found this really interesting maternal effect on larval survival, where if female oysters had experienced low pH 12 weeks before spawning, their larvae had much poorer survival in ambient pH conditions. And this was like one of the first plots I made as a grad student. And it was really cool to, one, make a plot in R, and two, have one with such an interesting result in it. Because the implication was that a long removed stressor could still impact larval survival. So returning now to this big question, right? How does DNA methylation influence intergenerational plasticity? First, we saw that low pH did indeed alter gonad methylation patterns, which was interesting for us to see because there were very few studies at that time that had looked at how reproductive tissue methylation responded to low pH conditions. So it kind of supported our idea that if methylation were to be heritable, we would see some interesting changes in the gonad. Our working idea right now is that differential methylation promoted homeostasis in adults, right? Promoted gonad development such that there were no differences of reproductive development between our pH treatments, but that same methylation may have been maladaptive for larvae. And the reason why I say that is because our working understanding of DNA methylation in invertebrates is that it reduces transcriptional noise, right? So reduces the amount of different kinds of transcripts that a gene produces. So we see female oysters reducing transcriptional noise. They're moved back to ambient pH conditions, and when larvae are spawned, they may inherit those same methylation patterns. So they may have lost promiscuous transcription that would be necessary for larvae to survive a really vulnerable life stage. So that reduction in transcriptional noise that was helpful for these female oysters may have in fact been harmful for larvae. One thing we're trying to do now is actually explore what happened. So I'm continuing to work with my collaborators at the University of Washington to look at the methylomes of larval oysters to understand how they relate to the methylomes of parents and how that relates to the conditions those parents experienced. So far, I've shown you how environmentally sensitive methylation could be a mechanism for both beneficial and maladaptive plasticity. We're going to shift gears here, and we're going to talk about some pretty big climate winners, the European green crab. So I'm going to talk to you a little bit more about the work I've been doing recently, exploring how plasticity impacts response to thermal stress in adult green crabs. So in case you haven't seen a green crab before, this is the European green crab. It is one of the most prolific non-indigenous species globally, and it is present on both coasts of North America. On the East Coast, on the West Coast, green crab presence again threatens ecological, economic, and cultural resources. They damage eelgrass beds, which are an important nursery habitat for juvenile fish and clams. I call them hungry, hungry hippos, because if it is smaller than them, they will try to eat it. So they can consume a lot of, this is particularly bad when we think about green crabs consuming a lot of clams seeded for commercial aquaculture. Additionally, yes, I'm so sorry. I just blinked out a little bit. I'm always slightly a little less useful after lunch. But right, they're damaging cultural resources. They're damaging aquaculture, like clam seeded for aquaculture, damaging eelgrass beds. All in all, not so great when we think about our native ecosystems. As their name suggests, European green crabs evolved in estuarine environments in Europe, with their native range spanning from Norway all the way down through Portugal and Morocco, right? So a really broad range of thermal conditions in that native range. Crabs made it to the east coast of North America in the early 1800s, and then from the East Coast made their way to San Francisco Bay in the late 1980s. From the Bay, they have spread northward through Washington, British Columbia, and as of a couple years ago, this map is actually out of date, green crabs have been found as far north as Southeast Alaska on the Pacific coast. But the interesting thing I want you to think about from this map is that, yes, they are spreading, but green crabs on the West Coast have undergone at least 2 genetic bottlenecks. One from the European range to the East Coast, and another from the East Coast to the West Coast, each time losing a substantial amount of genetic diversity. The only evidence of genetic selection that we have on the West Coast in this invasive range is a piece of the genome, and it's a set of genes that are inherited together, which I will refer to as a supergene. This supergene has several amino acid changing mutations in genes related to temperature. And the presence of the different kinds of supergenes that we see seem to be correlated with sea surface temperature. So in this plot over here, I'm showing you sea surface temperature along the x-axis, and then frequency of one allele of the supergene, the minor allele, on the y-axis, with different populations and different symbols. So the triangles are going to be our native range. The circles are going to be the East Coast, and then British Columbia in this study is our one West Coast population. And what I want you to take away from this plot is that there's this really nice linear relationship between sea surface temperature and frequency of one allele of that supergene, right? It seems to be a lot more common when sea surface temperature is warmer, as opposed to when sea surface temperature is colder. So based on this pattern, those of us who study this colloquially, are calling the supergene, what the supergene alleles, three different things, the three different flavors, right? So you can be a warm adapted homozygote, where you have two alleles of this, the two alleles of the supergene that are more common in our Portugal population, so in our warm temperature populations. You could be a cold adapted homozygote, so two copies of the supergene that is more prevalent in our cold water populations. And then you could be a heterozygote, so you've got one cold adapted allele and one warm adapted allele. So given this context, I was actually interested in understanding, how does a supergene impact physiology of individual crabs? Does that supergene have an impact on adult thermal physiology? Again, I had some hypotheses going into it. The first hypothesis was that individual level responses might vary based off of supergene genotype. And the second hypothesis I had was that differences in genotype distributions would then alter population level responses. So to start out, I did a 96-hour cold exposure experiment from crabs from both Massachusetts and Washington. And the reason why I chose cold temperatures for this experiment is, one, previous research has shown that green crabs have a really impressive, potentially boundless cold tolerance. They can survive in waters below 0 degrees Celsius. And experimentally, what has prevented us from really understanding where that bound exists is that water starts to freeze before a green crab heart stops beating. So I wanted to study this really impressive cold tolerance more. Additionally, as green crabs on both coasts continue to spread northward, up to Alaska, up to more Arctic Canada, I wanted to understand how they may be responding to ecologically relevant conditions they may encounter there. So I had a cold treatment of 5 degrees Celsius and a colder treatment of 1.5 degrees Celsius. Over the course of the 96 hours, we took multiple performance metrics, including survival, which I'll tell you right now, the cold temperature did not lead to mortality of any crab. And then we also performed writing response assays. And I really like writing response, one, because I work with a lot of undergraduates, and it's very simple to teach an undergrad how to do a writing response assay. You just take a crab and you flip it on its back, and you see how long it takes for that crab to flip itself back over. And because it's easy to do, easy to teach, it's also easy to take multiple repeated measurements of the same crab over a time series, right? It's not a time-intensive assay. So we looked at writing response, and at the same time, we genotyped these crabs to understand, were they warm-adapted homozygotes, cold-adapted homozygotes, or heterozygotes. So just before I get into the data, I wanted to thank all the really incredible undergraduates that I worked with that actually generated a lot of the writing response data, and very bravely sat in a cold room, stuck their hand in almost freezing water to flip crabs in the name of science. All right, so I'm gonna show you a lot of writing response data plots. And the way they work is all the same. We've got exposure time of the experiment on the x-axis, and then average time to write on the y-axis. And average time to write is gonna be in seconds. Our colder 1.5 degrees Celsius treatment is gonna be in a darker blue, and the lighter blue is going to be our 5 degrees Celsius treatment. First, we see patterns that we expected to see, right? Temperature, time, and their interactions significantly impacted writing response. And this is expected, right? Crabs are ectotherms, so their metabolic function matches the conditions around them. But we didn't see an impact of supergene genotype on their writing response. So if you're looking at this graph closely, perhaps you're noticing that there's only a handful of data points present in the box plots for the 1.5 degrees Celsius treatment. And earlier I told you that we didn't see any cold-related mortality. So what happened to these crabs? Well, at 1.5 degrees Celsius, we had over 50% of our crabs failing to write at all. So I wanted to look into this a bit further. So I've broken down our crabs that failed to write into crabs that had that were cold-adapted homozygotes, and then on the right, crabs that had at least one warm-adapted allele. So these are going to be our warm-adapted homozygotes and our heterozygotes. And I saw this trend where it seemed like if a crab had at least one warm-adapted allele, maybe it would fail to write a little bit more frequently than a crab that only had cold-adapted alleles. However, that was not statistically significant, so that inability to write was not actually associated with genotype. So this was data for the Massachusetts population. I'm going to walk you through the same data for the Washington population. Again, we saw very similar patterns. So writing response was dependent on time, temperature, and their interaction, but genotype had no bearing on average time to write. Interestingly, far fewer crabs failed to write themselves at 1.5 degrees Celsius when compared to our Massachusetts population. However, when we broke that down again, we did not see that genotype had any bearing on writing response in the Washington population. So far, our attempt to tie a population-level genotype association to individual physiology was not successful. So I was like, okay, perhaps it has no bearing on cold tolerance. What about heat tolerance? So I had a student working with me, her name was Julia Kelso, and she designed her own summer project to understand how genotype and plasticity influenced responses to multiple heat waves. So she had a subset of about 24 crabs from Washington, and she kept them at a control temperature of 15 degrees Celsius. On the first day, she exposed half of those crabs to a heat shock, a 24-hour heat shock of 30 degrees Celsius, returned all crabs to control conditions for 2 days, and then on the fourth day, exposed all of the crabs to a 30-degree 24-hour heat shock. The idea was to see if previous exposure to heat stress impacted subsequent exposure to that same stressor, and to see if genotype had any bearing on that. So again, I'm going to show you a time to write plot with day on the x-axis. Our red is going to be crabs that were exposed to that prior heat wave, so they were primed, and our gray is going to be our control, so no heat wave until the very end. So day 0, day 1, that first group experiences its first heat wave, and then on day 4, all of the crabs have experienced a heat stress. And what we saw was that priming conferred some sort of beneficial plasticity, where if we look at our control crabs between day 0 and 1 and day 4, right, there's this kind of heat stress associated slowing in writing response that we see in our control crabs that we do not see in the crabs that were primed to that heat stress beforehand. But that priming was not linked with supergene genotype at all. I think we did see some indication that crabs with at least one warm adapted allele were some of those crabs on the slower end of that distribution, but we just didn't have the sample size to have enough statistical power to be able to tell any, to see if that observation was in fact statistically significant. But for now, we can say that heat priming was not linked with supergene genotype. So, does the supergene impact adult green crab thermal physiology? We found that adult crabs can maintain broad thermal tolerance and plasticity in response to acute stressors, right? So 96 hours and shorter. But genotype had no impact on individual writing in any population or in any temperature condition. And this is because biology is complicated, right? It's really hard to link a population level genotype association with individual level responses. Adult thermal tolerance is likely the product of both is likely the product of both temperature shaping supergene distribution during larval recruitment and settlement, and phenotypic plasticity throughout life history. So I think that right now, adult crabs, looking at writing response in adult crabs may not be the best way to try exploring this association moving forward. But, if genotype isn't having a massive impact on thermal physiology in these crabs, perhaps it's time to explore some other functional mechanisms. And so that brings us to the last experiment that I'm going to talk about today, which is a 22-day temperature exposure I did with crabs from Massachusetts. And the goal with this experiment was to understand other molecular mechanisms that underpin this really impressive thermal tolerance that we see in green crabs. So stepping away from supergene genotype, thinking about functional genomics, things like metabolites and lipids. So for 22 days, we exposed crabs to one of 3 temperature conditions: a cold temperature of 5 degrees Celsius, a control of 13, or a warm temperature of 30 degrees Celsius. And across those 22 days, we looked at multiple different performance metrics, survival and riding response, in addition to oxygen consumption. So looking at the rate of oxygen drawdown for an individual crab at the temperature that they were kept at for the experiment. And to supplement these whole animal physiological metrics, we looked at untargeted metabolomics and lipidomics. So thinking about all the metabolites and all of the lipids present in an individual crab's heart tissue at the time of sampling. So for the rest of this talk, I'm just going to focus on the molecular results, but I'm happy to talk about these whole animal physiology results at any time. Okay. So the question guiding this last part is thinking about how does thermal stress influence crab physiology? Just a recap of those physiological results. We saw that crabs at 30 degrees were increased oxygen consumption, but may have been unable to maintain that response long-term because we saw a lot of mortality in our 30-degree treatment towards the end of the experiment. And we also saw that cold temperatures slowed physiological responses, but crabs showed some sort of acclimation response over time, where riding response at the end of that 22 days was a little faster, oxygen consumption a little faster. So what are the biochemical underpinnings of these trends? So we collected heart tissue from all 3 of our temperature treatments on day 4 and 22. I joke that this is my Disney villain era of science because I now have a freezer full of hearts. We then sent that heart tissue to the UC Davis West Coast Metabolomics Center for high-through mass spectrometry. So our analytical pathway is once we get the data back, we first conduct a multivariate analysis to understand the global impact of temperature and time on the metabolites or on the lipids. We then identified pairwise VIP molecules. So VIP stands for variable importance projection. In my head, this is a very important particle, right? So these are metabolites or lipids that were the most important for distinguishing different pairs of treatments from each other. Once we have our list of VIPs, we can then conduct a functional enrichment analysis to understand who those VIP are, what they're doing in the metabolic pathways, and how they may be helping a crab respond to thermal stress. So this is a PLSDA of our metabolomics data, where each circle is an individual crab's heart metabolome. And the metabolome is represented by 151 known metabolites. The colors correspond to temperature. So redder colors are 30, grayer colors are 13, bluer colors are 5, and the shades represent time. So the lighter shades are day 4, the darker shades are day 22. And we found that both temperature, day, and the interaction between the two impacted our global metabolite profiles. So the rate of metabolite reorganization was dependent on temperature and time. We can then start to drill down to some of those VIP. So we identified 29 VIP, so these are 29 individual metabolites that are responsible for distinguishing our 13-degree treatment from our 30-degree treatment at the end of the experiment. And a lot of these VIP were important in one pathway, valine, leucine, and isoleucine catabolism. So if you've ever taken any supplements, valine, leucine, and isoleucine are what are called branched chain amino acids, or BCAAs. Sometimes supplements are full of them. But BCAAs are really important for energy metabolism. That's why they're in those supplements. So enrichment of this process could signify that there was really high energy demand in our warm temperature treatment, right? Energy demand was likely outpacing supply of energy, so supply of those ATPs. crabs are moving very quickly, they're writing quickly, they're respiring quickly. There's just a lot of energy need at that hotter temperature, and we are potentially seeing that signature with the enrichment of this pathway. Looking at our VIP responsible for differentiating the 13 and the 5-degree treatment from each other at the end of the experiment, we identified 31 VIP, and a lot of these VIP were involved in arginine biosynthesis. So through a series of reactions, arginine can be converted into various intermediate compounds, at the end producing 14 ATP, right? And ATP are the energy currency of the cell. So similarly, arginine biosynthesis may be related to generating energy to respond to these cold temperature conditions. In cold temperatures, crabs also eat a lot less. The colder the water, the fancier the rock a crab is. They don't really move, they don't expend a lot of energy. So perhaps they needed to generate energy in other ways because they weren't feeding. So we think arginine may be used for energy production at 5 degrees Celsius, which could help sustain periods of low activity. I'm going to walk through the lipid results in a similar fashion. This is our PLSDA of our lipidomics data, made out of 415 known lipids. Interestingly, our lipid profiles significantly differed by temperature, but those responses were consistent over time, right? So that tells us that lipid reorganization is quick and immediate and consistent over a long period of time. And once again, I meant to point this out with the metabolomics data, but in both these data sets, we're seeing that this 30-degree treatment is farther away from the other two. So more unique lipids involved in the response to high temperature than response to low temperature. Once again, we can think about the VIP distinguishing these treatments from each other. We identified 130 VIP distinguishing the 13 from the 30-degree treatments. And a lot of these VIP were glycerophospholipids that were saturated. When we look at our VIP separating the control from the cold treatment, a lot of them were unsaturated glycerophospholipid. So what the heck is a glycerophospholipid and why do we care about them in the context of temperature? So glycerophospholipids are these compounds right here that make up cell membranes. We have the this hydrophobic fatty acid tail, and then the hydrophilic lipid head that points either out towards the outside of the cell or towards the liquid inside the cell, right? And so these fatty acid tails can change in their lipid composition in response to temperature. So when we have warm temperature, right? If we think about just butter, at warm temperatures, butter melts, right? Butters are made out of fatty acids, and if butter melts at warmer temperature, we don't want our cell membranes, our lipid tails, to lose structure and to melt in high temperatures. So to counteract this, organisms can add saturated fatty acids to their cell membranes. And these saturated fatty acids basically create these like kinks in the fatty acid tails so that they don't stack on top of each other as well. Thereby like preventing this membrane from like glooping out. So they can, other way around. Saturated fatty acids do not have kinks in the tail, so they can actually stack on top of each other really nicely and provide structure. Versus the other way around, at cold temperature, butter gets very hard and rigid. So to counteract the kind of rigidity that occurs with colder temperature, you're gonna wanna add unsaturated fatty acid to your lipid tails. So an unsaturated fatty acid has more of these kinks in it, so they don't stack on top of each other as nicely, so they provide a bit more flexibility in the cell membrane, which allows you to be more mobile in colder temperatures, right? So adding saturated fatty acids, which are flat tails stacking, unsaturated fatty acids, little kinks in the tail, adding flexibility, right? So that is why we think we're seeing these changes in glycerophospholipids occurring at those various temperatures. So to kind of recap, right, how does thermal stress influence crab physiology? We saw that metabolomic responses increased energy availability at both temperatures, but each temperature underwent a distinct process. We also saw that changes to membrane structure could underlie writing response acclimation, right? Making those cell membranes more fluid in cold temperatures or more structured in warm temperatures may have allowed that writing response acclimation to occur in our experiment. And then finally, we saw that metabolites and lipids had unique temporal roles in thermal acclimation. So just to kind of tie everything together, because I know I've been through a lot of things in this past 50 minutes, right? So when we were thinking about the oyster story, right, reproduction and intergenerational plasticity, we saw that environmentally sensitive methylation was a mechanism possibly for both beneficial and maladaptive plasticity. And we made some connections between methylation and physiology. But my next steps are trying to make some connections between DNA methylation and the transcriptome, so what genes are being expressed, and then connect that to physiology across multiple generations. When we're thinking about green crabs, we've done a lot of exploration at the various mechanisms that could be underlying their broad thermal tolerance, thinking about genotype, thinking about metabolites and lipids, and how all of that may underlie whole animal physiological changes. But our next step is to try to add some layers of gene expression into our experiments to understand more of the connections between genotype, gene expression, and these metabolic pathways, as well as thinking about different life stages that may be important to study. So with that, I just want to thank you for your attention, and I'm happy to answer any questions.
I have a question to start it out. I'm just curious, you're using a lot of these omic techniques. Are you having your undergrad students helping out with these analyses?
That is the goal, yes. So Santa Clara University is an undergraduate university, so my lab is comprised solely of undergraduate students. The goal is to get everyone really excited about computational biology. And to not, at least at the very least, not see a computing cluster as an enemy. But eventually the goal and the idea is to have the undergraduate students work a lot more with the transcriptomic data sets and metabolomic data sets that we're working on generating at the moment.
Excellent talk, thank you. I'm curious about whether you've got any thoughts about experiments in the future where you might manipulate diet with different lipids or different kinds of food to see if that can impact the outcomes of some of these climate stressors.
Yeah, that's a really great question. Thinking about diet manipulation and how it impacts response to stressors. I have not waded into that territory yet, but it is something that I've been thinking about. So when doing crab experiments in particular, we feed them what's called Hikari crab cuisine. It's like a crab pellet meal that you could buy at any pet store. And I've always thought about how feeding like live food may differ from those pelleted foods and how that might impact response to these stressors. So it's something I've definitely considered in the back of my head, but haven't tested yet. I think that it would make a really great undergraduate project if there was an interested student, for sure. I'm also like working with a recirculating seawater system, so there's only so much I could do. Like I, at the moment, don't have the capacity to grow, at least if I do want to do shellfish stuff, like grow strains of algae to feed oysters. But yeah, something to consider.
Thanks for your talk. So I was intrigued that the crabs from Washington, it seemed like more of them were able to write at that really cold temperature than the Massachusetts crabs. And I was wondering if you had thoughts on that, because it seems like, if anything, the Massachusetts crabs should see colder temperatures, at least during the winter.
Yeah, definitely. Why are Washington crabs better at writing at colder temperatures when Massachusetts crabs, or crabs on the East Coast in particular, definitely routinely see much colder temperatures than crabs in Washington in the winter? I have been thinking about this, and I'm wondering if Massachusetts crabs fail to write more as like an energy conservation strategy right? Because these crabs are likely encountering colder conditions, perhaps they may have energy conservation strategies like not writing very quickly or writing at all in order to preserve limited energy in colder conditions. One thing to note about our writing response studies is we have been working with like a threshold where if crabs don't write within 90 seconds, we mark them as not writing. I think if I were to do this experiment in the future, I wouldn't keep a threshold, or I wouldn't keep maybe that low of a threshold. Might try to bump that threshold up or not keep it at all to see like if we had given them like 3 to 5 minutes, would they have righted themselves? And then is that maybe a more robust metric that you can compare across populations?
When you get into genomics and transcriptomics, the real challenge is annotation. And I guess, especially with genomics, I'm curious about the oyster work. When you identified these methylation sites and sorted them according to genes and non-genes, did you incorporate all the genes that were identified as containing a coding sequence, even though it might not have been able to be identified? Or did you focus your annotation on identified genes in the genome?
Yeah, so when doing when trying to understand where differentially methylated loci are located in the genome, do I just look at genes that are annotated, or do I look at genes that we know are genes but maybe don't have an annotation? The latter, right? So at least for the genomic location portion of it, if we know it's a gene sequence, even if we don't know what that gene does, right, that was interesting for us to look at. When it came to functional enrichment, right, that's where annotation starts to matter a lot more, right? It's hard to understand the potential implication of a differentially methylated locus in a gene where we don't even know what that gene does. There are some ways that you can analyze the data to potentially cluster by expression, or cluster by methylation level genes with similar functions. And then if you've got some unannotated genes in that cluster, you can maybe start to get at or start to hypothesize about their function a lot more. I will say for oysters, we're really lucky. A lot of the Pacific oyster genome in particular, is very robust, very well supported, pretty decently annotated. Versus the green crab, right, we don't have a robust genome yet, especially with the metabolomics enrichment analyses. There were a lot of things that I had to be a lot more conservative with or really look twice at, just because the annotation workflow for green crabs, so a species without a lot of annotations, right, robust annotations, and then metabolites, which are way less supported in non-model organisms. I think that's where the tricky part was.
Hey, this is actually a good follow-up question to that question. And we had talked about this like so briefly when we chatted earlier, but how are you dealing when you're annotating or at least comparing your methylation data between your treatment and controls, dealing with the heterogeneous nature of the C. gygis genome? Yeah.
So how do I deal with inherent variability in the oyster genome when doing my analysis? I tried to reduce that from an experimental standpoint. So all of my oysters that I used in my experiment were half siblings, right? So that itself reduced a lot of genetic variability. I think it'd be interesting and important to kind of consider methylation analysis or methylation experiments across different stocks of C. gygis, but that wasn't within my capacity at that moment. So try to reduce genetic variability from the outset, and then when you're aligning your sequences to the genome, right, I try to strike a balance between allowing for more mapping and being conservative in that mapping, and I think that's kind of where I was like, we'll do the best that we can programmatically, and then just move forward with what we feel comfortable and confident with.
Can I ask a follow-up question? So, Zala, Is that so? Sorry. So for the mapping, did you just do like a general, I guess, genome, or did you like do a de novo genome from your like half sibling population?
No, we took the Roslin Dragos genome.
Okay, cool.
Yeah.
Thank you.
Although I think there's a newer one now, but yeah.
Yeah, but this one was good.
Or our student questions, great. Oh, yeah, Kara, go ahead and hand that one. Okay.
This is really interesting. Thank you so much for presenting. I was wondering what you might, from your research, what do you think people in conservation and restoration might be able to do with it? And additionally, do you think there's any room for genetic modification to kind of slow or make these crabs more susceptible to and like temperature changes so they don't spread as far?
Yeah, so answer your question in two parts. The first part was, what are the kind of conservation implications of the work that I do? I think that I might just speak to implications broadly. So for my PhD, for the oyster stuff, I worked a bit more closely with oyster hatcheries. And I think that oyster hatcheries are always interested in kind of optimizing their brood stock, optimizing what gets outplanted, thinking about like what is going to be the most resilient to disease or to various environmental conditions. So in that sense, I can kind of see like exploring stress hardening or exploring multi-generational impacts of stress to be kind of important knowledge when we're thinking about like, can we then take this information and try to do something with it to improve aquaculture practices. As far as the grain crab goes, and to answer kind of the second part of your question, could we do some sort of genetic modification with grain crabs to maybe stop their spread, assist with invasion management? That is something that people are thinking about. Thinking about using like a CRISPR gene drive to, similar to what they kind of try to do with like Anopheles, like malaria mosquitoes, to like sterilize them. I think there's a little bit more work that needs to be done to get to that point, but it is certainly an intervention strategy that people are considering. I don't think I know enough about the implications of that intervention strategy, both positive or negative, to be able to have like a strong opinion about it at the moment. But I think it is interesting to consider, especially when we think about like green crabs are so well established on the East Coast. They're pretty well established in California. Like they're moving north, right? And it seems to be that like once you have an adult population, right, they're kind of there. So I think it would be an interesting intervention tool to think about for sure.
Yay, this was such a cool talk. I really like the super gene crab experiments, but I just have like a question about, I haven't thought much about methylation and like thinking about how it drives like transgenerational plasticity. I'm just wondering if organisms are able to like change their methylation in response to acute stress, why don't they change it back once it's better?
Yeah, that's a great question that kind of touches at like the transient nature of methylation modifications and epigenetic modifications broadly. The simple answer to your question is we don't really understand it that much, right? I don't think we have a good enough handle on the kind of wash in, wash out dynamics of methylation modifications or epigenetic modifications, which is why like the kind of legacy effect that I saw was interesting. I think that is something that I am hoping to explore more to kind of understand, right, like at what point are different patterns established, not just at the methylation level, but thinking about gene expression level, right, like small molecule level, at what point do those effects go away? And what's maybe the lag time between like the start of a stress and the end of the stress and then the end of like a potential molecular machinery change. Yeah, short answer is we don't know, but it's interesting.
Awesome, very exciting.
I have another question about methylation. I don't know very much about it, but I was wondering, where did you get your, I guess the methylation chapter was oysters? Were your oysters like already lab acclimated? And was there any chance that
the methylation changes could have been due to being in a lab versus being under heat stress? And like, do you account for, was there some sort of like control that you accounted for in that when looking at like the differential methylation?
Yeah, great question, right? So like captivity effects essentially on like molecular responses. So I ran this experiment out on an oyster hatchery where these oysters had been for quite a bit. So the hope was that like they were kind of used to the hatchery, right? We ran like an acclimation period of them in the actual tanks that we were using, in the bags that we were using as well. But these were hatchery stock oysters. It's, I think, definitely something to consider if we were like pulling wild stock out and then bringing them into a hatchery. But these were like hatchery reared, hatchery raised oysters that had been in the hatchery for at least like a few months by the time that we had gotten to them. Yeah, but I think captivity effects are something that I always think about, but that's also why we always tend to have like experimental control, right? Like, can we isolate, like if there is some sort of captivity effect, it's likely uniform, right? So any differences we see between our control and something else, we can hopefully attribute to our experimental modification. Yeah.
That's kind of a follow-up on the last question, and I think you kind of touched on it, but do you think that the results would be different if you, in this hypothetical situation where you're able to take one plot of water inside the ocean with like non-captive oysters, do you think the results would be different, or would they be the same? I worked with captive fish before, and a lot of times their behaviors don't have that kind of fear, I guess, of humans. So I don't know.
Yeah, great question. So like what you're thinking about is like a outplant-ish mesocosm study. I think, right, in a manipulative laboratory experiment, right, we're able to control a lot more. I think that if we were to do something out in the wild, there'd be like maybe not just the condition that we're interested in manipulating, but like other conditions might be changing as well, right? Like food availability might be changing, temperature, dissolved oxygen, salinity, all that. Aerial exposure, right, if you're working with shellfish that are outplanted on a tideland. So to answer, but also not answer your question, maybe things would be different, but I think there would just be a lot of other changing factors that might need to be accounted for when you're actually thinking about what you're interested in and what you actually want to see modifying the phenotype that you're interested in.
I think our time is up, but I'm sure Yamini would take a few more questions afterwards, so please join me in thanking her for a wonderful summer.